Bismillahirrahmanirrahim. The topic is oligosaccharides. We already studied about the monosaccharides, they are the basic carbohydrates, and the different properties and structural configuration of those monosaccharides. Now we'll come to the oligosaccharides, the second category of the carbohydrates, and these are the carbohydrates made up of monosaccharides. When monosaccharides combine, they form the oligosaccharides. Oligosaccharides then can be divided into two categories, that is the disaccharides and polysaccharides. Disaccharides are the oligosaccharides made up of the combination of the two monosaccharides. When two monosaccharides combine, a bond between them is formed, and their bond is known as glycosidic linkage or glycosidic bond. Glycosidic bond combine the two monosaccharides and they form one oligosaccharides or disaccharides. And when many monosaccharides combine, many glycosidic linkage form between them, and that is known as polysaccharides. Now, first of all, we will discuss the oligosaccharides of the disaccharides of oligosaccharides. Disaccharides, as already mentioned, that these are the carbohydrates that when two monosaccharides combine, they form the disaccharides. Disaccharides, especially the general formula with the C12H22O11, these are less sweet in taste as compared to monosaccharide and less soluble in water. Example are given here is the sucrose, maltose and lactose. First of all, we will come to the formation of the sucrose and some properties of the sucrose. Sucrose is a disaccharide made up of alpha glucose and beta fructose. Alpha and beta, what is the difference between the alpha and beta? That if the hydroxyl group at carbon number 1 position lower, it will be alpha. If hydroxyl group at carbon number 1 position upper, then it will be beta. So here the glucose is alpha and the fructose is beta. So when glucose and fructose combine, they form the structure, the glycosidic linkage. One group release hydroxyl and the other release hydrogen. This hydroxyl and this hydrogen, carbon number 1 of glucose and carbon number 2 of fructose, release water, one hydroxyl, one hydrogen, it release in the form of the water. The carbon number 1 will attach with the carbon number 2, and this attachment or this bonding is known as glycosidic bond or glycosidic linkage, and the water will be released. So this is a disaccharides because two monosaccharides combine, alpha glucose and beta fructose. Sucrose is very important, especially in the plants. Plants make their food in the form of the glucose and then convert it into in the form of sucrose because it is less soluble and less cannot be hydrolyzed easily, and this can be a best carbohydrate for the movement of the food from of plants inside the plant from one area to another area. So this is a transport disaccharide, which can be when the material is transported, it mostly change into the form of the sucrose. The next is the maltose. Maltose is another disaccharides, but it is made up of alpha glucose and alpha glucose. Two glucose, they form alpha 1,4 glycosidic linkage, means carbon number 1 of alpha glucose and carbon number 4 of another alpha glucose. These two combine, they release the water, and then a glycosidic linkage between the carbon number 1 carbon number 4 is formed, and the two glucose give the maltose. It is also known as malt sugar because it is present mostly in the fruits, and this is also produced during the malting. And what is malting? Malting is the breakdown of starch with the help of the enzyme amylase, and during that process maltose is produced. So that process is known as malting, which is very important in the production or industrial brewing process. Another one is the lactose, which is known as milk sugar, and because this is found in the milk, and especially in the milk of the animals, also in the milk of the humans. And this lactose is formed by the combination of the monosaccharides beta galactose and beta glucose. Beta glucose combined with the beta galactose, carbon number 1 and carbon number 4. One release hydroxyl, another release hydrogen, and they form the water, while the 1 and 4 glycosidic linkage is formed between galactose and glucose, and this structure is now then known as lactose. And this is also very important because on hydrolysis is give galactose and glucose, and also the milk sugar is very important for the growth of the living organism, a very important form of the living organism. Now the polysaccharides that are made up of many monosaccharides. When more than 2 monosaccharides combine, they form the 4 polysaccharides. Polysaccharides then contain the 2 categories, homopolysaccharides and heteropolysaccharides. When the same monosaccharides combine, sometime all the glucose combine, sometime all the fructose combine, sometime all the galactose combine. The same units combined, they are known as homopolysaccharides, and when the different monosaccharides combine, they form the heteropolysaccharides, and they are non-soluble, that are very complex and having no taste. So that is the reason that when the carbohydrate is needed to store, that is changed into polysaccharides because they are not easily converted into monosaccharides, and they are not soluble easily. The first one example here is given is the starch, and that is in the combination of the all, you can see here that all alpha glucoses, all these are the alpha glucoses combined with each other with the alpha 1,4 glycosidic linkage. Each glucose is attached to another glucose with the alpha 1,4 glycosidic linkage. So the monosaccharide unit is the alpha glucose, and this found in the 2 form. One is the amylose and the other one is the amylopectin. What is the difference between the two? Amylose is a linear chain. It is found in a linear chain, while amylopectin is found in branch structure. The 2 chains are then further attached with each other. Amylopectin, each glucose is attached with 1,4 alpha glycosidic linkage, and then the 2 branches are attached with the 1,6, means the carbon number 1 of this branch is attached with the carbon number 6 of this branch, and forming the bond alpha 1,6 linkage, glycosidic linkage with each other. So this is the difference between the amylose and amylopectin. Amylopectin found in branch form, and it is found in linear chain form. Another example of the polysaccharide is the glycogen. Glycogen is also known as the animal starch because this is the polysaccharide which is normally have the same structure like the amylopectin, but here the structure is more branch. Like we can see that just like this the branches occur. Amylopectin have to some extent less simple structure, that less complicated structure as compared to glycogen. Glycogen is a more complicated structure. The same 1 to 4 linkages of the glucose, alpha 1 4 linkages of the glucose. All these are the glucose unit attached with each other with the 1 and 4 glycosidic linkage, as well as the branches 1 to 6 linkages found, but this is more branch as compared to amylopectin. And glycogen is known as animal starch because the animals, when needed to store the carbohydrate, they convert the simple carbohydrate into glycogen, and glycogen is then stored in the bones or in the blood or in the different areas of the body. Next is the cellulose, the most abundant carbohydrates on earth. That is the basic component of the plant cell wall. We already know that the plant cell wall is made up of cellulose. So this is the most abundant carbohydrate on earth, and this is formed by the combination of the different glucose beta 1,4 glycosidic linkage, beta glucoses. Like we already studied that the starch and the glycogen, these are the derivatives of the alpha glucoses. Here the beta glucoses linked with each other, beta 1,4 linkages. Like this is the beta glucose, and these two beta glucoses are linked with 1 to 4 linkages, one carbon number 1 attached with the carbon number 4, releasing water and forming glycosidic linkage. So 1 to 4 beta linkages form the cellulose, and cellulose is the carbohydrate which form the structure of the plants, maintain the structure of the cell wall. Next is the last one is the chitin, the second one abundant carbohydrate on earth, and this is the mainly the structural part of the arthropods skeleton, exoskeleton of arthropods, and this is the only one form of the carbohydrates that it consists of the glucose, but as well as glucosamine and N-acetyl group. N-acetyl and glucosamine group are attached to them, making it more complex, forming the beta 1,4 glycosidic linkages. Glucose are attached with the beta 1,4 glycosidic linkages, as well as the N-acetyl group is attached with it, so they are known as N-acetyl glucosamine linkages, and this is very important in maintaining the structure, especially the skeleton of the arthropod, as well as it forms the fungal cell wall. Fungal cell wall consists of chitin, which is the second one abundant carbohydrate of the earth.